Yeah, hello everyone. I'm Garyk. I'm a PhD student with Brian He, and I'm excited to talk to you all about EVO2, our DNA language model. So, as the audience will know, biology is encoded in DNA. Whether something happens at the level of protein or RNA or regulation, the information is likely encoded in the DNA sequence. DNA is a foreign language to us. So you can know what comes next if we say to be or not to be. But what about ATG? It's probably a repeat that's coming up. So models can learn from language, from data, right? And human language was a foreign language to AI models before pre-training. And pre-training through network prediction has enabled models to learn many tasks. And intuitively, you can see how wanting to predict just the next token or word may help you learn something about grammar, semantics, or the world, or various other things. Because if you want to be able to predict what comes next accurately, you want to learn representations and circuits that can generalize to new text. What about for DNA? So DNA encodes the genotype, it's the information. And through various mechanisms, this will be transformed into an organism's phenotype. Natural selection then acts on this level, altering the types of genotypes we observe. So in this way, information flows back about mechanism into the genotype. And a quick example to kind of illustrate this, we can think about a case where we might have two genes that are very distant from each other, but they are made into protein and they interact. So even though they're distant on the DNA, they interact physically in the cell. And that interaction may be between some amino acids on the proteins. And if one of them mutates, the other one, the interaction may be destroyed. So the other one may have to compensate and mutate. And across long evolutionary time, we can observe this kind of covariation among distant sequence positions. And this tells us something about the biological mechanisms at play. And so this kind of intuition has been used by various statistical and machine learning models. And hopefully it helps illustrate how language modeling on DNA could help tell us something potentially about disease, about biochemistry, about RNA and protein structure, or about even systems biology. We already know that these approaches can work. So the best predictive models of proteins often leverage this kind of evolutionary signal. AlphaFold famously uses a multiple sequence alignment, which is explicitly looking at many sequences from different organisms. And follow-up research showed this MSA is crucial for getting a good guess of the protein structure that can then be refined. And we've previously showed that protein language models, despite being single sequence models, also exhibit characteristics of these evolutionary types of models. So we have great models of protein sequence, but how can we learn from the DNA directly to capture the different scales and modalities inherent in biology? So in biology, you will be interested in individual elements and genes that might be tens or hundreds of nucleotides long, up to many millions or even billions of nucleotides for the entire human genome. So these different modalities and scales interact. They're all written in DNA. So we hope that models of DNA can help capture them and model them together. Unfortunately, genomes are uniquely hard to model. They have a lot of repeats, and we mean a lot, like the majority of the human genome. These will be simple, such as the same token or base pair over and over again, or complex, such as gene systems that jump around and copy themselves across the genome. And intermixed in this will be essential information on critical genes. Genomes are also multi-scale. So it can take thousands of base pairs or more to encode just one single gene, which maybe is analogous to one word in language. but any one base pair mutation might be able to change, and if it changed, destroy this gene's function or create a new function. So you need to be able to model very long sequence, but you also need to be able to model individual small changes. So EVO2 is a long context DNA language model trained across all domains of life that tries to address some of these challenges. To train EVO2, we create a new machine learning architecture to improve throughput and quality for this byte tokenized data. So this architecture, striped hyena 2, is what we're calling a multi-convolutional hybrid. It combines 3 different types of hyena layers with self-attention and alternates these layers throughout the model. Through ablations, we show that this improves perplexity on our byte tokenized data, and it also improves throughput, both at short and long sequence lengths. So the intuition here is that the different layers will be specialized to different tasks. The short and medium layers are parametrized explicitly, analogous to CNNs that you might be familiar with, while the long layer is similar to the original hyena. This is an implicit convolution. And when we scale this up, we see up between 20% improvement against a optimized transformer baseline in terms of throughput at short sequence, and this improves with sequence length. But critically, we're improving a throughput both at short and at long sequence lengths. So there's a companion paper that we've preprinted, which details the algorithm and systems work that enabled EVO2 training. And I recommend you all to check that out for details on algorithms that were used to enable the training up to 1 million base pairs.
Garyk?
Yeah.
I think in your paper you say something like a 3 times speed up for 1 million long context and a 40 billion parameter model or something like that.
Yes.
So is this, I thought we mainly would care about memory efficiency, like that we can't even train our model with longer context if we don't use hyena. So is that wrong?
So the speed up, so with the speed up is in part due to it is due to the quadratic nature of attention, but fitting the transformer at long context is not necessarily an issue if you split it very aggressively across many GPUs. So
Okay,
there are
then what are the things that you're splitting across GPUs in your model? Like you shard your model, right?
yes,
How do you do it?
Yeah, so there's so for the 7 billion parameter model, the sharding is tensor parallel, but only during the context extension, so only at longer context lengths. And then for the 40 billion, there's both tensor parallel and then context parallel sharding. So there's a ring attention implementation, and there's also an analogous implementation for the hyena layers. And this is the new algorithm mentioned in the preprint, in this companion preprint. So there's both the sharding the model weights and sharding the sequence context.
Okay. And when you shard model weights, is it like the feed forward layers, or is it like you have different feed forward layers, or is it that you put each layer onto a different GPU? No, that sounds bad, because then we would no, you do both, right? You shard across feed forward layers, and that's what you call like token sharding or something like that. And then you also shard the individual layers of the model, or what are the two things?
Yeah, so I think you're referring to pipeline parallel, which is splitting the models layer by layer across different GPUs.
Okay. And that's
So
not what you do, right?
yeah, we split it. You
Mhm.
can think of it as like column wise. So the different columns in the weight matrices are split. Yeah.
Okay, the different columns in the weight matrices. Um, so essentially each, you know, but you also said you put, okay, for the 40 billion model, you also put different tokens on different GPUs.
Yeah, but that's yeah, and that becomes more of an issue at the longer and longer context lengths.
Okay, okie dokie. Thanks. Oh wait,
Yes.
now one more thing. How many shards do you have per like for your 40 billion model, 1 million context length, into how many pieces do you split your model?
Yeah, good question. Off of my memory, it's going to be it's like either I think 128 or 64. But the problem is that this is stage wise, right? So I don't remember because at each stage we tune it. Jerome was
Yeah,
handling this tuning, but yeah. So all of the configs are public, and we can check this out and reference those. But I think
okay.
one of those two numbers should be correct.
So each GPU handles 1/28th of a batch or of a single sample.
Yeah.
Okie dokie.
Yeah, so we have our architecture, and now we need a data set. So we create Open Genome 2, which is a data set of non-redundant DNA from across all domains of life, totaling nearly 9 trillion DNA base pairs. And we find that data composition, weighting, and augmentation are critical for training on DNA, likely due to those challenges that I mentioned earlier. So during our training, we end up focusing primarily on regions proximal to genes, so coding and non-coding sequence within 10,000 base pairs of some annotated gene. And we find that this, through a series of small-scale ablations, we find that this improves performance and scaling on downstream tasks. And in our paper, we actually conduct ablation at 7 billion parameters. And this shows a very big difference of training on full genomes versus training in this more focused way, where the focused way is able to do better, both on coding and on proximal non-coding, significantly better performance. So Evo 2, similarly to many human language models, is trained in phases. We first focus on a shorter context pre-training phase, during which the majority of tokens are consumed. And then we do a long context extension in mid-training. The intuition is that you get better representation of functional elements, and then you learn how to compose it in long sequence. And this has been shown to improve quality in various other works also. So during training, we see that our loss improves both with model scale and with context length, showing the model is using the longer context to improve its prediction of the DNA. And we verify this with the needle in the haystack evaluation to show that EVO2 is able to pick out this 100 base pair homologous DNA sequence from anywhere in the 1 million base pair long context length when it's filled with a random DNA background. So this suggests the model is at least able to recover sequences from even very far away. So
Sorry,
now, yeah.
what does this score here mean?
Yeah, good question. So this is a little bit arbitrary. This is the delta logits from the categorical Jacobian. So we're not actually generating the sequence. We are mutating the needle and then observing the model's prediction on the query. So the scores are a bit arbitrary, but generally they're if they're above one, they reflect like a strong coupling for the model.
Okay, so why would a haystack length, a smaller haystack length be harder?
Yeah, I agree. I don't think that the the differences in the numbers are super meaningful. We probably could present this with a more binarized evaluation, and I think that might make more sense. Yeah, I'm not sure why they are lower. I didn't look into it, but my yeah sense is this is an arbitrary unit that is kind of it's nonlinear, right? It's kind of like a classification.
Okay. And then you're already talking about nice results now, but about the training, do you also tell the model somehow what type of sequence it gets as input? Like sometimes you have mRNA, sometimes you have DNA, sometimes you have um, I don't know, something else, and you always tokenize it in the same way, or the tokens always look the same as far as I understand. Um, and then how do you tell the model?
Yeah, for the majority of these data types, we don't tell the model additional information especially during the shorter context pre-training. There are some special tokens for data sets that have artificially created discontinuity. So these these splice sites, for example, where we're taking pieces of the introns, but only short overhangs, and then joining it with the next splice site. There we place a special token and loss is not passed on that special token. But yeah, we do not we do not include information on the specific data set as a conditioning. What we do is during the context extension for the various genomes, we include the phylogenetic information as a special conditioning, a series of tokens. Loss is not passed on those. And these are included only during the second phase of training to help the model.
So what is this information? Like which is it an animal or what?
Yeah, it's
What does it mean?
information on it's so the actual form that this takes is a string containing the phylogenetic information starting from broadest to most specific. So it'll go from, species and upwards.
Uh-huh. I see. Okay, so you're able to, for example, generate something that is conditioned on being in a mouse.
Yeah.
Okay.
Yeah. So
I'm sorry.
yeah.
Can you go back to the scores, the matrix slide? Yes. So, I don't do tool development, so I don't have the comprehensive context here. But I'm trying to understand the score. So, the higher score meaning you have a better
Sorry, can I quickly
ability.
interrupt you? So these scores are like you look at the logits that your model outputs after you're making a mutation in your inputs, and then you compute this categorical Jacobian from it, which there's a neat paper that you can look at just if you search categorical Jacobian. But I think this figure is not not a very important result. So let's
Okay.
rather continue and look at more important results.
Okay.
All right. Yeah, so we have this model, and now we want to see what it can do. So one of the things that would be interesting to tell is how the likelihoods out of the box, zero shot, reflect fitness. Because ideally, this DNA language model could predict fitness for DNA, RNA, and protein sequences. So we first look at whether this model captures biological
Sorry, I had a
information.
quick question. So the previous plot, is that a schematic or is that signifying something real data?
But yeah, this is a schematic.
Okay, so I tried to like figure out what that was in your paper and I couldn't figure out because um, I thought this was pairwise likelihood changes or something. Well, what are you trying to convey here?
So that there's a high dimensional sequence space. So yeah, it's not actually two dimensional. The idea is just there's different sequences and you can score them.
Okay, and WT is one particular wild type.
Yes.
Okay, so it's a schematic though. That's what I was
Yes.
confused
Yeah.
with. Yeah. All right, good. Thank you.
So the likelihoods, so we look at the likelihoods and how they change around gene starts for different species. So this is going from eukaryote to prokaryote, looking at coding genes. And immediately you can see that the model is picking up some patterns that match what you'd expect. So there's a lot of attention paid to the first codon in the protein. And then for the subsequent codons, the third position, which is known as the wobble base, is the likelihood changes are smaller, and this reflects lower conservation, lower functional importance of that third base due to redundancy in the codon table. We also see in the 5' UTR, this conserved region in prokaryotes also matching what we might expect for the 5' UTR. So we then look at how EVO can predict gene essentiality. So this is using experimental data from different species where different genes have been mutated, and then there's growth screens to assess the fitness of those organisms. So the zero-shot likelihoods of EVO2 can predict the gene essentiality for these phage and bacterial genes. And so while we trained on eukaryotes and prokaryotes together, EVO2 is able to predict for prokaryotes and then also for eukaryotes. So we repeat a similar experiment focusing on a new long non-coding RNA essentiality data set from human cell lines. And we find that EVO2 is able to accurately classify these as essential or non-essential zero-shot. So we then focus on variant effect prediction for human mutations because we have a lot of data on human mutations and we care about predicting them accurately. And specifically, we care about being able to predict different types of mutations, which a DNA language model should enable. So, and this, just to summarize in case people are unfamiliar, the way that we're doing all of these calculations is by looking at the log likelihood that comes out of the model, so multiplying all of the logs together, and then comparing this for the mutant sequence and the wild type sequence. So we can first look at ClinVar, which is a large database of expert annotated disease mutations. And we see that EVO2 is able to predict both coding and non-coding, as well as substitutions or more complex insertions and deletions. So for coding mutations, for substitutions, there's other models, they're very good. And EVO is competitive, but not as good. But for non-substitutions, EVO does well on coding and does well on non-coding. And when we put all these mutations together, EVO also has good performance, showing it's not just able to predict these mutations separately, but it has some calibration between them, at least for these high effect size pathogenic mutations. We then focus on BRCA1, which is a cancer-associated gene for which there are good deep mutational scanning data sets. We see EVO2 can predict the coding and non-coding effects. So for coding, EVO is very good, although not quite as good as AlphaMissense, which was especially trained on human population data. But EVO is state-of-the-art for non-coding variants. And so these together show a very strong baseline for this model that was trained across all species with no special preference given to humans, that zero-shot it's able to predict these coding and proximal non-coding mutations accurately. And putting coding and non-coding substitutions together, we see EVO2 performs very well, showing that calibration that I was mentioning, which is something we really wanted to see from our DNA language model. And like with other models, fine-tuning can further improve the accuracy. So when we use EVO2 embeddings and train a classifier on top of it, we can improve the performance on this BRCA1 allele.
Garyk,
Yeah.
can you explain the data here a little bit more? There's this clean var, and later on the splice var thing. And in clean var, like what are the readouts? So what different type of readouts do we have? And yeah, why does or what is, for example, the mechanism in which a non-coding change might affect such a readout?
So for ClinVar, this is a data set of mutations in patients that have been found to be associated with some disease. So there's pathogenic and non-pathogenic and different scores among those two, but these are human expert labeled. So clinical geneticists will consult various sources of information, including looking at experimental data from research papers and, of course, looking at patient samples, and they will, and maybe looking at some computational methods, and they will try to find causal mutations that cause a genetic disease, and those will then be nominated as being pathogenic. So ClinVar is looking at mutations that are actually associated with some disease, and the coding or non-coding, there may be many different mechanisms, right? It might be, and there's the non-SNVs, which are insertions and deletions, but most of these mutations will be kind of very high effect size because they're causing a disease that can be traced down to a single mutation change. SpliceVar-DB, I don't have on my slides, but this is in the paper, and that's a data set curated based on various experiments trying to find mutations that alter splicing. So while ClinVar is kind of a phenotype effect on the patient level, SpliceVar-DB is affecting splicing. So experimentally verified mutations that if that mutation happens, the splicing of the gene changes.
And we don't care about how it changes, we just care about that it changes.
Yeah, we think that yes, in that zero shot benchmark is just that it has altered the splicing.
It will be interesting to try to somehow use the model to change splicing in a desired way. Okay, and then you have your disease. You're just saying, oh yeah, right. How do you construct your negative mutations?
Yeah, in this data set in ClinVar, there are negative mutations.
I see.
And I guess the good thing about those is that they will appear similar to the, in order to get to that point, they had to look similar enough to be nominated. So they're likely rare, right? It's not like a extremely common mutation likely. But there's other benchmarks that will use common variants as baselines, for example.
Do you have an understanding of the data for how, like if I make a random mutation, how likely is it to cause the specific pathogenicity if you make that mutation inside of the same gene?
I'm not sure I understand the question.
So you take your gene, you have a mutation. That mutation is in a particular gene, and it causes a disease. And if you now take the same gene and make a random mutation in it, then how likely is that to still cause the pathogenicity?
Yeah.
So if you just have the same gene.
Right. I think that is somewhat captured by the gene essentiality style benchmark, where there you are saying you're knocking out the gene maybe in different ways, but you're seeing how bad knocking out the gene is on the organism, let's say. But I guess to try to answer this question, there will definitely be plenty of mutations that do not cause pathogenicity, even if they hit a essential gene. So they might be a gene that can cause disease, but some, of course, many mutations may have no effect, and some may be a partial loss. And so the model, this kind of benchmark does require you to predict the importance of the specific mutation. You can't just rely on the gene level importance.
Yeah, I understand the benchmark. I was just wondering about the biology, how how often it is important that there's like a specific mutation that changes the okay, if I knock out the gene versus I make the specific mutation, how different will the effect be, right? Because it might be that if you have this mutation, then your protein does something stupid that is pathogenic. But if you just knock out the protein, then well, you're still fine if it's not
Yeah.
an essential protein.
Yeah, absolutely.
But
I think it's a bit of it is still an open question. And probably if we could answer it, we probably wouldn't have to build DNA language models if we knew ahead of time. Right. But I think I think that's a good question. And there's some research that suggests that a large number of the pathogenic mutations are LOF-like. That they're similar to partial or full loss of function, but there's definitely a fraction, probably a smaller fraction, that create a new stupid function, like you were saying.
Yep.
All right, yeah, great questions. And let's see, I see some questions in the chat.
Yeah, is some of this data tied to RNA-seq data to determine expression in specific tissue types or to discern 5' UTR patterns to determine this?
We don't use any RNA-seq data in training. It's all DNA sequence. And we also haven't looked at expression prediction in as a downstream either. So yeah, not something we've looked at. And Francisco, oh yeah.
If you don't use any RNA-seq data, then how does your mRNA stuff work, or your mRNA experiments?
All right, so yeah, I guess implicitly there was RNA-seq used to annotate the genomes, but we go from the annotated genomes to construct the mRNAs. We don't look at RNA-seq data sets.
Okay, so you go ahead and you know that this snippet in your DNA is mRNA and it's spliced together like this and that. Or is mRNA also spliced together?
Yeah.
Okay. And then you try another. Okay.
Yep. Yeah, so the data is all from the annotations. Yeah.
Sorry, just a question on the previous slides. So I wonder how sensitive it is for this result. I know like when you actually do the inference, you can either going from the whole DNA sequence, or you can chunk it to basically use a small set to do this prediction. So how sensitive this result is this to the choice of the chunk size that you choose?
Yeah, good question. We use primarily 8,192 as our window size for the zero-shot prediction. We find this works well, but we don't find big differences with using much longer or slightly shorter sequence, although it seems shorter does do worse if you do shorter than 8,000.
Okay, sounds good. Thanks.
Yeah, thanks, Boyang, for the question. And for future questions, let's put them in the chat. And I'll also cut down on my number of questions so we can get through all pieces of meat that we want to cover. And then, yeah, we're sure we're not missing something more important. All right, let's continue.
Okay, so hopefully we've shown that there's this impressive zero-shot prediction ability. And this question remains, what is Evo2 learning? What's in this black box? And there's recently been lots of progress on interpreting large language models with mechanistic interpretability. So as you may have seen from Anthropic, people can identify interesting concepts and features in these large language models, and then even change the model behavior by affecting those specific parts of the model. And so we teamed up with Goodfire to train sparse autoencoders on Evo2, and then use the resulting autoencoders to identify biological features, focusing on annotation and discovery. So specifically, what we do is we look at different activations in the sparse autoencoder, and then we use genome annotations. We find features that are enriched for various annotations, and then see if those are robust. So we find that Evo2 has learned features at protein, RNA, and DNA levels. So for RNA and protein, we see features which correspond very well with transfer RNAs, with alpha helices, and with beta sheets across many different types of proteins. And so here's some plots mapping those alpha helix, beta sheet, and tRNA features onto predicted structures for those proteins. And you see very good concordance. Evo's also learned DNA regulatory motifs, such as motifs associated with known transcription factors that are critical and conserved. There was also a phage-associated feature identified. So this is very interesting because it's a single feature that corresponds to prophages, CRISPR arrays, and also phage-derived genes. So whether this is an entire phage that has integrated in a bacterial genome, whether it's small chunks of phage in the CRISPR spacers, in the CRISPR repeats, or whether it's just single genes that were originally evolutionary from phage, but then were taken and put elsewhere by the bacteria and maybe repurposed. There's this feature that fires on all of them. Evo2 also learns features corresponding with genome architecture in both eukaryotes and prokaryotes. So these are features for CDS, introns, as well as the start and end of exons. And we run it on the woolly mammoth genome, which was not in our training data, and see this good correspondence between the features and genome organization. I'm pausing for some questions in the chat.
Maybe what about potential interactions between genes that are far apart or even in different chromosomes?
This is not something that we looked at with the interpretability, but I think it would be definitely interesting to look for features that might be linking distal elements.
Okay.
So in mech-interp research, there's been a finding that there's features that identify bugs and errors in code. And we actually find a feature that identifies bugs in DNA code. So this feature does not fire for synonymous mutations, which don't have a functional effect. It has very little firing for non-synonymous mutations or even stop codons, which both can have a high effect, but it's specific to frame shifts, which are mutations that throw the coding sequence out of frame so they can significantly alter the protein and make it go and gain a function like Hannes was talking about earlier. So this might be a type of mutation that can cause that. And there's this feature that across many different genes corresponds to frame shifts. And the interesting thing about this is it's kind of a complicated feature. You have to know that you're in a coding sequence, and then you have to know that that this mutation, this, let's say, deletion or insertion that has thrown the rest of the protein out of shape, that has happened. So you have to have an understanding of what a good protein will look like, and that to identify this mutation. So I think it's an interesting result, and you can look at these features online on a website and scroll around various genomes if you want to play with them.
So how does let's go with mine first. How does your feature mining work, or this contrastive feature mining, I think you call it? Contrastive feature search. Yeah, sorry, did I just miss that? I think I just missed it. Then let's just continue.
All right. Yeah, so I think this is, these results are exciting because they suggest the model, although pre-trained just on DNA sequence, you're learning interesting features that match to our understanding of biology without ever telling the model that a protein exists or a data sheet. So this is a generative model, so we can also use it to generate. So for this, we first look at prompting, generating various sequences. We start with the human mitochondria and prompt with 3,000 base pairs, generating the rest. So on the left is a synteny plot of various mitochondria. In the middle, you'll see the wild type generation, and above and below, the wild type sequence, and above and below, you'll see evo-generated mitochondria, annotated with a mitochondrial annotation software. So evo2 generates mitochondria with the correct number of tRNA and coding genes and RNAs, and they maintain synteny while demonstrating significant diversity, including sometimes things like rearrangements or changing of ordering or splitting of genes. And you can presumably tune this kind of diversity based on your hyperparameters and your specific task of interest. We look at the proteins that were generated, fold them with AlphaFold 3 and co-fold them together, and we see that they fold to this expected structures. The amino acid percent identity here, which is between 75 and 90%, is quite high for protein engineering, but mitochondria are very conserved, and these are very sensitive proteins. So if you compare human to zebrafish homology for many of these proteins, it'll be around 80%. So it's actually a fairly diverse as far as animal mitochondrial proteins. So we next look at generating prokaryotic sequence, starting with M. genitalium, which is a prokaryote with a genome of 600,000 base pairs long, which fits within our context window. So we prompt with the start of this genome, and then we generate on, and we compare ourselves with EVO1. We see a significant higher number of the genes that were generated have PFAM hits. So these are genes that have some homology to real genes. And we see a distribution of gene lengths, which matches that of the natural wild type sequence. We then co-fold our genes using EM-SEM-fold and look at the secondary structures. And we see that the distribution of the secondary structures for EVO2 generated genes is close to that we would expect based on the wild type. So this is exciting because we're getting meaningful generations of various genes, even when generating very, very long sequences. We apply the same method for a yeast, this time generating an entire chromosome, and then compare this to the wild type. We see it, and we annotate with yeast genome annotation tools. We see EVO is generating genes, introns, promoters, and tRNAs, and many of these genes, most of these genes being homologous to natural genes. So the levels generated are still below wild type for many, especially for tRNA and also for genes. But encouragingly, we do see the secondary structures match that of wild type. So we've demonstrated that EVO2 can generate with prompts, and then we wanted to see how it can be used in, well, let's pause for the question.
Okay, so there's this Nature paper that generates cell type specific enhancer sequences. So they demonstrate that they can turn on, or they can generate enhancers that turn on certain genes in different cell types. Does your phylogenetic thingy or your phylogenetic token sequence also capture the cell type?
No, we don't capture the cell type, and I think that it would take some thinking, I guess, because we're trying to include the whole genome and also have it at long context. Because it's, all the cells have the same genome, and yeah, some parts are active in some cell types. So you could think of some strategy to do it, but yeah, our tokens
And
are.
what does cell type even mean, I guess?
That's
Okay.
true. Change over the time of the organism's life.
Okay. Yeah, maybe the model can't be so easily used for generating cell type specific stuff.
Right, or maybe it can, because, and so I think we'll cover this a little bit right now. So I think that one idea is to use this kind of base generative model of the DNA and combine it with maybe a cell type specific guidance model, right? And do inference time search. So we look at epigenetics and specifically using Informer and Borzoi as guidance while using Evo2 as the generation. So in this way, you can design, let's say, chromatin accessibility to a specific pattern where you're just sampling from Evo2 and then guiding with a attack-seek predicting model. So here we see we're generating along the sequence. The predictions are in blue, and in yellow, there's the desired shape of the attack-seek peaks. So this is inference time search. We're just generating and then searching over generations to find ones that match the desired design. As you search more, you get better designs that match your criteria. So if you only generate very little, you won't find designs that match what you're looking for. But if you sample more, you can. And so we see this as an interesting combination of generative DNA models with specific applications. So similar to a lot of other work that looks at in AI that looks at how to use inference time compute and scoring functions, we think that Evo2 can enable various generative tasks by being combined with different classifiers and reward functions. So hopefully we've seen that Evo2 has learned general capabilities by pre-training on just DNA. It can predict variant effects for DNA, protein, or RNA. It can discover biological concepts just through pre-training and then extracting the activations, and it can generate realistic DNA. We hope that Evo2 can enable a kind of app store for biology where it can be combined with different task-specific methods and allow us to, by reasoning on DNA, to combine different modalities. So please check out our preprint and our GitHub. Evo2 is fully open source, including the training code, the various ablation experiments and configs, the inference code and data. And it's only possible through this very large and amazing collaboration. So I'd like to thank all of my teammates. And I'd also like to thank NVIDIA, who helped make training Evo2 possible. So I thank them for their vision. All right,
Okay.
so let's take some questions.
Thanks, Garyk. First meta question. How much, oh no, not so meta. How much compute did you get from NVIDIA? Can you say anything about that?
We describe in our, I think we describe in our paper, so the Evo2 40 billion was trained on 2,000 GPUs.
Okay. And do you just get access to some Slurm cluster that they set up?
Yeah, we were training on NVIDIA's cluster.
Okay. Cool. Then any questions in the chat? What is the idea of genome generation evaluations? For example, do we want the sequences to be conserved or diverse? It feels like the expectation would be confusing for different positions in the genome. Yeah, anything to say?
Yeah, I think this is a good question it's something we encountered, especially if you want to generate something for human or for some other animal genome. It's maybe hard to know how to even annotate it with a single sequence. A lot of annotation tools, which you might want to use to try see, did you make a gene and do you think it will splice correctly? A lot of these tools use homology and similarity to databases, and they use RNA-seq. So if you're generating a gene that is completely new, it may be very hard to even annotate it. And then there's the question of the diversity versus quality trade-off, where we have a hard time assessing the quality of generations that are too different from nature. So I think this is probably something that will improve over time as we have more people trying different things, and especially with some sense of what works in the lab.
Okay. Then what do you think is the most important downstream task that needs long context?
I think that generating long sequences, needs long context, and I guess it's like most important is maybe hard to
Okay,
say.
just
Yeah.
pick from your top 3 a random one.
Yeah, I'll just go with generating long sequences. I think that this is something where if you want to make a coherent generation that does not, that maybe can be less redundant or something, it's good for the model to remember what it's made and be able to reference that.
Sure, but now you say generating long sequences, but what is the actual use of like you can generate them and look at them, but yeah, what would be the actual use?
Well, it depends how long we're talking about, but I think for sequences of tens of thousands, low tens of thousands, there will be interesting uses in biology. If you're talking about a plasmid or a similar construct, we don't have to be talking about a million base pairs, but if you're able to generate 10,000 base pairs and encode something in it, I think this is
Yeah, and I think
imminently usable.
I think Vera is now just asking about what is one compelling example in biology. Like you talked about plasmids just now, where you like maybe this doesn't work perfectly yet, but let's say we wait another 2 years or so, improve the model a bit. What would you want to do with the long sequence generation capabilities?
Well, 2 years, I guess for very long sequence capabilities, I think that maybe this is a more longer term question. But for something like, yeah, for just make a circuit that has a certain logical operation, right? You encode several genes, they're regulated in a certain way, they regulate each other, and this is enabled with a DNA language model in part, maybe combined with other approaches. I think that that's something that would be possible, because right now, let's say you want to like do this epigenetics task that we talked about. So you want a gene that is conditionally open in some context. And if you just copy natural elements and try to do this, you can introduce a lot of problems, right, in your sequence by being highly homologous to other pieces of DNA. So having a language model to diversify sequence and keep it not natural, this is very useful for various synthetic biology things. And then I guess I see now the second part of this question was just generally the point of a big model. I think Evo 2, it's a big model, and this is what lets it do well across many species. We evaluate a lot on human because we care about humans and we have a lot of data on them, but we did not treat humans any differently during training, right? So these zero-shot performance would hopefully be consistent across many different contexts, and I think that that's much harder to do with a small model. You have to specialize more.
can I use your model or your 40 billion model with a 40 gigabyte GPU?
Not the 40 billion, but you can use the 7 billion, which is which I like, which I think is a good model.
Okay.
Can I do maybe a follow-up on my question, please? Just
let's go.
Thank you. So my question would be, would it be meaningful to use, for example, Evo2 40 billion model for a task such as a functional genome annotation? Or in this case, a smaller model with that is trained on smaller context length would be better?
I think, yeah, I think that genome annotation likely requires an understanding of, I mean, depends on how similar the genome is to genomes you've already annotated or sequenced. But hopefully, if you have a DNA sequence, and you have no idea what it is, you put it in Evo2, you should have a sense of where the genes might be. And we've done this from predicting from the embeddings. We've done this with the SAEs, the MECINTERP strategy. So there's different ways you can imagine using language models for annotation, like you were suggesting. And we have some encouraging, initial results in that direction. So I think that this is definitely a possible application, because I mean, you can think of Evo2 as compressing all of this data and finding some connections between it and sharing parameters between it. So I think it should help when you're applying it to some organism that you're not really sure about. It might be different from the other ones you've seen.
Thank you, but not maybe just like the whole, like for example, given like a shorter sequence of like, I don't know, 400 base pairs. And then the question is this enhancer or not? Would it, like how if I used Evo2 for 40 billion, would it be better than a smaller model? I mean, did you have any ablations on that? That's the question.
I mean, I think this depends on the task, right? Like in identifying where an enhancer is might be relatively easy to learn, and if you train something very specific, yeah, I'm not sure. I think it would depend on what you're using it for.
Okay, thank you very much.
Okay, then do you know, or can you tell me what CPG islands are, and do you model them do you model something like DNA methylation?
Yes, we do not use DNA methylation data, and we do not have any, we only use the DNA sequence. So it's a generative model on DNA, and we don't include any molecular tracks. You can think about this as a way to train across evolution. We don't have functional genetics data, functional genomics data from many species, but we do have their DNA.
Yeah, I mean, you could expand your vocabulary by 2, like 2 8s, and now just have also methylated
cytosine,
DNA.
yeah, methyl cytosine and hydroxymethyl cytosine. Yeah, you could add that in, definitely.
Anyway, what are CPG islands? Do you know that?
Yeah, clusters of high density CG. So they should be in the model somewhere, because they're just CG rich areas that are usually near promoters. But there are other places in the genome and in repetitive areas too. But they're probably in the model somewhere, I would guess. Just haven't been pulled out.
Okay, sounds good. Then I guess we have some questions about the mechanistic interpretability stuff after all. Could you, like, yeah, I guess some people, including me, didn't get it. How do you label the SAE features?
Yeah, it's a bit of a it's a bit of an art. I'll go back to or actually maybe I can just go back to the schematic. The idea, the automatic portion is that there is a annotation. So we need to have an annotated DNA track. This will mark which positions on the DNA match a concept. So let's say it's protein coding gene. And then the SAE runs along, you have the activation for each feature along that same sequence. And so now you're just calculating your precision recall for your annotations in versus out, right? Versus the feature and how it's firing. And so that's the more automated portion of the feature search.
Okay, so you end up with a threshold basically for every feature and then telling what it indicates.
Yeah, you end up with a measure of the difference in activation, like in and out of
yeah, okay.
Yeah.
Okay. Then could you describe more how the phylogenetic tokens look like?
Yeah, they're in the they're on the GitHub. We have a helper function that can help you pull it for a species you're interested in. So if you're interested in actually using it, that's what I would look at. But I think that the format, I think, is called green genes. It is simply the ordering of the phylogenetic information starting from broadest to most specific, and it's in text separated by like underscores and semicolons. But I think it would be most helpful if you look at either in the paper or in the GitHub to get the precise function that will create these for you.
Okay. Then shows emergent yeah, let's maybe have this as the last question. Can you talk again a little bit about or explain in more detail the mitochondrial generation evaluations?
Yeah. So the mitochondria were annotated using, wrong way. The mitochondria were generated using a 3000 base pair prompt. So this is from 0 to 3000 for the human reference mitochondria. We then generate with some hyperparameters and save all of the generated EVO2 mitochondria. We annotate them using mitoz and then look at the proteins and the RNAs. So on the right side in this table, it's just quantifying the counts for tRNAs and protein genes and rRNAs. So that gives us a sense that it's completing the mitochondria with an appropriate set of genes. And then on the left, this is a plot using Lewis for you. It's looking at the synteny. This is kind of qualitative, right, to see that the order is what you would expect. And then we look at homology of the various generated genes using BLAST-P and the homology of the whole sequences using BLAST.
Okay, so you basically you evaluate how well do you recover the original sequence. If your model were to just memorize it, it would perform well in these evaluations.
Well, it would perform well on the counts of genes, but for example, on this synteny plot, right? The thing we're excited about is that it is not memorizing, that there are some changes even in terms of introducing new genes that maybe exist in other organisms but are not in humans, or duplicating or splitting. And then the BLAST homology kind of seeks to make sure that it's not just memorized. But yeah, there's this trade-off between diversity and our ability to assess the generations.
Okay. Maybe I lied, and we'll have a look at some more questions after all. Thank you for the amazing talk. Indeed, I concur. Will you be testing any of your generations? Will you evaluate any of your designs in the wet lab experimentally?
Yeah, we hope to, but I think more on this to come in the future.
Okay, that sounds good to me. Then, any last words from your side before we call it a day?
Thanks for hosting me. I think that DNA language models are an exciting way to take advantage of all of this great sequencing that everyone has done over the past 20 years. We have all these sequences, let's put them together, compress them, learn these cool representations and see what that unlocks. So I hope you're all excited as I am for the future.
All right. I am definitely excited, and thanks for pushing this stuff. Cool. Then thanks, Garyk. Thanks everyone for joining, for the nice questions, and see you all friends of the sun next week.